بسم الله الرحمن الرحيم. دكتور أحمد النجار, Lecture of Histology and Cell Biology Department. Today we will discuss and explain the cardiac muscle and smooth muscles. Cardiac muscle fiber are striated and involuntary muscles. What is the site of the cardiac muscles? They are present in the wall of the heart, in myocardium, in the origin of pulmonary veins, superior and inferior vena cava. Again, cardiac muscle fiber are striated and involuntary sites of cardiac muscles. They are present in the wall of the heart, in myocardium, and in the origin of pulmonary veins, superior and inferior vena cava. Light microscopic of cardiac muscle. Size. Cardiac muscle is smaller than skeletal muscles. Shape. Cardiac muscle fiber are branching and anastomosing. Each cardiac muscle fiber is formed of a chain of cylindrical cells joined together by intercalated disc. Cardiac muscles fiber are branching and anastomosing. Each cardiac muscle fiber is formed of a chain of cylindrical cells joined together by intercalated disc. Cytoplasm of cardiac muscle. Cytoplasm acidophilic and has glycogen and lipid granules. Nucleus of cardiac muscles. Each cells in cardiac muscle fibers has single central oval nucleus, rarely binucleated cells. Again, light microscope of cardiac muscle. Size. Cardiac muscle is smaller than skeletal muscles. Shape. Cardiac muscle fiber are branching and anastomosing. Cardiac muscle cells joined together by intercalated disc. Cytoplasm acidophilic because high rich of glycogen and lipid granules. Nucleus. Each cells in cardiac muscle fiber has single central oval nucleus, rarely binucleated. Direction of the fiber of cardiac muscles. Fibers in cardiac muscle run in different directions. Run spirally around each heart chamber in three directions: transverse direction, longitudinal direction, and oblique direction. Thicker in ventricle with strong force. Direction of the fibers. Fiber in cardiac muscle run in the different direction. Run spirally around each heart chamber in three directions: transverse direction, longitudinal direction, oblique direction. Thicker in the ventricles. Electron microscope of cardiac muscle. Cytoplasm show cross-banding with alternating light and dark band, but less distant than skeletal muscles. Again, cytoplasm show cross-banding with alternating light I band and dark A band, but less distant than skeletal muscle. Sarcomere has the same structure as in skeletal muscle fiber, but sarcoplasmic of cardiac muscle fiber contain numerous myofibrils, numerous mitochondria. T-tubules are more numerous and larger. Sarcoplasmic reticulum is less abundant. Glycogen granules, lipid droplets, lipofuscin pigments, Golgi complex. Again, the sarcoplasm of cardiac muscle fiber contain numerous myofibrils, numerous mitochondria due to 40% of cytoplasmic volume, indicating need for aerobic respiration. T-tubules are more numerous and larger than in skeletal muscles. Sarcoplasmic reticulum is less abundant. Glycogen granules, lipid droplets, lipofuscin pigment, Golgi complex. The cardiac muscle fiber has diet system, not triad as in the skeletal muscle, formed of one terminal tubules and one terminal cisternae of sarcoplasmic reticulum. Again, the cardiac muscle fiber has diet system, not triad. Triad is in skeletal muscles. What is the diet of cardiac muscle fiber? Diet formed of one terminal tubules and one terminal cisternae of sarcoplasmic reticulum. The T-tubules are white and surround the myofibrils at the Z-line. Differ from skeletal muscles. T-tubules in the skeletal muscle are at the A-I band junction, but in cardiac muscle, T-tubules at the Z-line. 2. Sarcoplasmic reticulum is less developed than in skeletal muscle and form one narrow terminal cisternae along one side of the T-tubules to form the diet. Again, T-tubules are wider and surrounded myofibrils at the Z-lines. Sarcoplasmic reticulum is less developed than in skeletal muscle and form one narrow terminal cisternae along the side of the T-tubules to form the diet. Intercalated disc. What about intercalated disc? Intercalated disc is the attachment site between cardiac muscle cells. Intercalated disc consists of transverse part and longitudinal part. Transverse part of intercalated disc cross the cardiac muscle fiber at right angle to its longitudinal axis at Z-line. Longitudinal part of intercalated disc is parallel to long axis of cardiac muscle fiber. Transverse part of intercalated disc contain 3 junctions. 1. Fascia adherens. 2. Desmosome junction. 3. gap junction. Fascia adherent junction numerous. What is the importance? Importance of fascia adherence is connecting actin filament of adjacent cells into the sarcolemma. Desmosome macular adherence importance is bind the individual cells to one another, preventing separation of cardiac muscle cells during regular repetitive contraction. Gap junction provide ionic continuity between adjacent cardiac muscle cells. This exchange makes cardiac muscle fiber to behave as syncytium. Longitudinal parts of intercalated disc contain one junction. It's called gap junction. Gap junction. Rich in gap junction. So, gap junction present in transverse part and longitudinal part, but rich in longitudinal part. Again, intercalated disc is the attachment site between cardiac muscle cells, which formed of transverse part and longitudinal part. Transverse part cross the cardiac muscle fiber at right angle to its longitudinal axis at Z line. Longitudinal part of intercalated disc is parallel to long axis of the cardiac muscle fiber. Transverse part of intercalated disc contain three junctions: fascia adherens, more numerous, desmosome, and the gap junction. Fascia adherens connecting actin filament of adjacent cells into sarcolemma. Desmosome bind the individual cells to one another, preventing separation of cardiac muscle cells during regular repetitive contraction. Gap junction in transverse part of intercalated disc provide the ionic continuity between adjacent cardiac muscle cells. This exchange make cardiac muscle fiber to behave syncytium. Longitudinal part rich in gap junction. Regeneration and growth of cardiac muscle. There are no satellite cells, so capacity of regeneration is poor. So any damage of cardiac muscle fiber heal only by fibrosis. Again, how the cardiac muscle regenerate? There are no satellite cells, so the regeneration is poor. So any damage of cardiac muscle fiber heal only by fibrosis, which lead to myocardial infarction. Cardiac muscle fiber grow only by hypertrophy, increase in size. Again, growth of cardiac muscle only by hypertrophy, increase in size. Describe the structure of the wall of the heart. Formed of three main layers: epicardium, myocardium, endocardium. Again, the structure of wall of the heart formed of three main layers: one layer epicardium, two layer myocardium, three layer endocardium. The whole covered by pericardium. One epicardium outermost layer. It is visceral layer of pericardium formed of simple squamous epithelium outer part. A layer of connective tissue contain coronary blood vessels and fat cells. Blue color, fat cells, blue color. Again, epicardium is outermost layer. It is the visceral layer of pericardium, formed of simple squamous epithelium, outermost layer, and the inner layer of connective tissue contains coronary blood vessels and fat cells. 2. Myocardium. Myocardium is middle layer, much thicker in the ventricle, especially left ventricle, more than atrium, formed of cardiac muscle fibers spirally arranged around each chamber, separated by loose connective tissue, contain blood vessels. Again, myocardium is middle layer, much thicker in ventricles, especially left ventricles, more than atrium, formed of cardiac muscle fiber spirally arranged around each chamber, separated by loose connective tissue, contain blood vessels. 3. Endocardium. Endocardium, the inner layer, lines the chambers and cover the valve, formed of 3 layers: simple squamous endothelial layer, the inner layer, 2 subendothelial connective tissue layer, middle layer, 3 subendocardial layer, outer layer. Simple squamous endothelial layer is the inner layer. Subendothelial connective tissue layer contain elastic and collagen and smooth muscle fibers. Subendocardial layer of loose connective tissue contain blood vessels, and a very important fiber is called Purkinje fiber. What is the Purkinje fiber? Purkinje fiber, conducting system of the heart. It is continuous with connective tissue of myocardium. Again, subendocardial layer of loose connective tissue contain blood vessels and Purkinje fiber. Purkinje fiber is conducting system of the heart. It is continuous with connective tissue of myocardium. Moderator band. The conducting system of the heart began from SA node, then AV node, then moderator band. What is the moderator band? Moderator band is the right branch of AV bundle of His. It is formed of Purkinje fiber, which are a special modified type of cardiac muscle cells. Purkinje fiber differ from cardiac muscle fiber in 1. Larger in size, with 1 and 2 central nuclei. 2. Cytoplasm of cardiac muscle cytoplasm of Purkinje fiber is pale and vacuolated due to its high content of glycogen, which pushing the cytoplasm into stellate-shaped strand. 3. Cytoplasm contain fewer myofibrils that are present mostly peripherally. 4. More rapid conduction of impulse of Purkinje fiber, rich supply with nerve. Purkinje fiber are surrounded by connective tissue rich in blood vessels. Again, The conducting system of the heart began from SA node, then AV node, then AV bundle of His and its branches. This is called moderator band. Moderator band is the right branch of the AV bundle of His. Is formed of Purkinje fiber, which are special modified type of cardiac muscle. Purkinje fiber differ from cardiac muscle fiber in larger in size, with 1 and 2 central nuclei. Cytoplasm is pale and vacuolated due to its high content of glycogen, pushing cytoplasm into stellate-shaped strand. 3. Cytoplasm contain fewer myofibrils that are present mostly peripherally. Purkinje fiber are more rapid conduction of impulse due to rich supplied by nerves. Purkinje fiber are surrounded by connective tissue rich in blood vessels. Smooth muscle are non-striated and involuntary muscles. What's the site of smooth muscle of the body? They are present in the wall of the blood vessels, wall of the various viscera, as in digestive system, respiratory system, genitourinary system. Development of smooth muscle fiber is from the undifferentiated mesenchymal cells in perinatal life or pericyte cells in postnatal life. Again, development of smooth muscle fiber in perinatal life from undifferentiated mesenchymal cells, in postnatal life from pericyte cells. Light microscope of smooth muscles. Size smallest. The size of smooth muscles is smallest. Shape: smooth muscle fiber are elongated, physiform, spindle shape, non-branching cells. Nucleus of smooth muscles: single, oval, central nucleus, occupy the widest part of the cells. Cytoplasm: acidophilic, non-striated, containing dark patch of dense bodies, scatters in the cytoplasm, surrounding by basement membrane. Again, a light microscope of smooth muscles. 1. Size smallest. 2. Shape: smooth muscle fiber are elongated, fusiform, spindle shape, non-branching cells. Nucleus: single, oval, central nucleus, occupying the widest part of the cells. Cytoplasm of smooth muscle: acidophilic, non-striated, containing dark patch of the dense bodies, scatters in the cytoplasm, surrounded by basement membrane. Electron microscope of smooth muscles. Sarcoplasm of smooth muscle contain 3 sets of filaments. 1. Thick myosin filaments. 2. Thin actin filaments. 3 intermediate filaments. Thick myosin filament and thin actin filament not arranged in sarcomeres. Intermediate filament formed of desmin and vimentin protein, which form the cytoskeleton network. Other containing of sarcoplasm of smooth muscles: rough endoplasmic reticulum, Golgi apparatus, and the mitochondria. They are connected by gap junction. Again, electron microscope of smooth muscles. Sarcoplasm contains 3 sets of filament: thick myosin filament, thin actin filament, intermediate filament. Thick filament and thin filament not arranged in sarcomeres. Intermediate filament formed of desmin and vimentin protein, which form the cytoskeleton network. Other containing of sarcoplasm: rough endoplasmic reticulum, Golgi apparatus, mitochondria. They are connected by gap junction. Structure of smooth muscle fiber by electron microscope. 1. Myofilaments. Myofilament, they are differ from that of skeletal muscle in 1. The preparation of actin to myosin. Actin is blue color, myosin is brown color. The preparation of actin to myosin is greater than in striated muscles. 2 difference: the myofilament are not organized in parallel, myofibrils are not arranged in sarcomeres. Instead, they are grouped in bundle with oblique orientation to the long axis of the cells. 3. The thin myofilaments lack troponin complex. They are attached with one end to the dense body, while the other end is free, interdigitating with the thick myofilaments. Again, myofilaments is the structure of smooth muscle fiber, appear by electron microscope. They differ from that of skeletal muscle in 3 things. 1. The preparation of actin to myosin is greater than in striated muscles. The myofilament are not organized in parallel, myofibrils are not arranged in sarcomere. Instead, they are grouped in bundles with oblique orientation to the long axis of the cells. 3. The thin myofilament lack troponin complex. They are attached with one end to the dense body, while the other end is free, interdigitating with the thick myofilaments. How contraction in a smooth muscle fiber occur? It is based on the sliding filament mechanism. Sliding of the thin filament over the thick filaments lead to generate a pull on sarcoplasmic and subsarcolemma dense bodies. Then shortening of the smooth muscle fiber. Again, contraction in smooth muscle fiber, it is based on the sliding of filament mechanism. Sliding of thin filament over thick filament generate a pull on sarcoplasmic and subcirculemma dense bodies. Then shortening of smooth muscle fiber, then muscle contraction. Structure of a smooth muscle fiber by electron microscope. Cavuli. Definition of cavuli: vesicular invagination from the sarcolemma of smooth muscle fiber. Again, cavuli is vesicular invagination from the sarcolemma of smooth muscle fiber. What's the function of cavuli? They are regarded as the counterpart of T tubules, which are absent in smooth muscle fiber. Again, two structures of smooth muscle fiber appear by electron microscope. Cavuli is vesicular invagination from the sarcolemma of the smooth muscle fiber. What is the function? They are regarded as the counterpart of the T tubules, which are absent in smooth muscle fiber. Three structures of a smooth muscle fiber appear by electron microscope. Sarcoplasmic reticulum. Sarcoplasmic reticulum is poorly developed and consists of narrow sarcotubules that are often associated with the cavuli. This association allows transmission of the electrical stimulation of the cell surface to the interior of sarcotubules with the release of calcium and the initiation of the muscular contraction. Again, structure of a smooth muscle fiber by electron microscope showing sarcoplasmic reticulum. Sarcoplasmic reticulum is poorly developed and consists of narrow sarcotubules and associated with cavuli. This association allows the transmission of electrical stimulation of the cell surface to the interior of sarcotubules with the release of calcium and the initiation of muscular contraction. Other organelles and inclusion which appear by electron microscope in the structure of a smooth muscle fiber. They are mainly located in the poles of the nucleus. They include mitochondria, Golgi complex, few rough endoplasmic reticulum, and ribosomes, centrioles, glycogen granules, and fat droplets. Again, other organelles and inclusion in the structure of a smooth muscle fiber include mitochondria, Golgi complex, few rough endoplasmic reticulum, ribosomes, centrioles, and glycogen granules, and fat droplets. Structure of a smooth muscle fiber appear the gap junction by electron microscope are found between the adjacent smooth muscle fibers What is the function of gap junction? Facilitate transmission of the impulse for contraction from one smooth muscle fiber to another. Again, what is the importance of gap junction? Gap junction are found between the adjacent smooth muscle fiber and can facilitate transmission of the impulse for contraction from one smooth muscle fiber to another. Function of smooth muscles. 1. Contract involuntarily under control of sympathetic and parasympathetic nerve of the autonomic nervous system. 2. Senses of type 3 collagen, elastin, proteoglycan. Again, what is the function of smooth muscles? 1. Contract involuntary under control of sympathetic and parasympathetic. 2. Senses of type 3 collagen and elastin and proteoglycan. Growth and repair of smooth muscles. Growth occur by proliferation by mitotic division of smooth muscle fiber and hypertrophy, increase in size. Repair of smooth muscles is by no satellite cells. Proliferation by mitotic division of the smooth muscle fiber or contractile pericyte cells from the wall of the blood vessels, particularly in the repair of the vascular smooth muscles. Smooth muscle fiber respond to increased load by hypertrophy, increase in size, and hyperplasia, increase in number. Again, growth and repair of smooth muscles. Growth occur by proliferation by mitotic division of the smooth muscle fiber and hypertrophy, increase in size. Repair of smooth muscle fiber is by no satellite cells, by proliferation of mitotic division of smooth muscle fibers or contractile pericyte cells from the wall of the blood vessels, particularly in repair of vascular smooth muscle. Smooth muscle fiber responds to increased load by hypertrophy and hyperplasia. Hypertrophy, increase in size, hyperplasia, increase in number. Comparison between skeletal, cardiac, and smooth muscle fiber. Contraction. Skeletal muscle is voluntary, cardiac and smooth muscle involuntary. Striation, distinct striation of skeletal muscle, less distinct striation of cardiac muscle. Smooth muscle non-striated shape. Skeletal muscle cylindrical, unbranched. Cardiac muscle cylindrical, branched, and anastomosis. Smooth muscle fusiform, single, unbranched. Compare between skeletal muscle, cardiac muscle, smooth muscle. Skeletal muscles, fibrous shape and nuclei, single, cylindrical, multinucleated cells. Cardiac muscle, aligned cells, branching, single, central nucleus. Smooth muscle, small, fusiform, single nucleus. Striation, smooth muscle unstriated, cardiac and skeletal muscle striated. T-tubules, skeletal muscle striate at AI band. cardiac muscle diate at Z disc, smooth muscle cavuli, smooth endoplasmic reticulum, well developed diet in skeletal muscle, less diet in cardiac muscle, irregular in smooth muscle. Junction not present in skeletal muscle, intercalated disc in cardiac muscle, gap junction in smooth muscles. Contraction control in skeletal muscle, troponin C bind calcium. Cardiac muscle, troponin C bind calcium. Smooth muscle, calmodulin bind calcium. Innervation, skeletal muscle motor, cardiac and smooth muscle autonomic. Response to load, skeletal muscle hypertrophy, cardiac muscle hypertrophy, smooth muscle hypertrophy and hyperplasia. Capacity of regeneration, skeletal muscle limited by satellite cells, cardiac muscle poor regeneration, smooth muscle good, involving mitosis of muscle cells. Thank you.